Every human cell carries 2 meters of DNA that is carefully compacted inside a nucleus of only 10 micrometers in diameter. We now know that the way our DNA is folded and packed inside nuclei plays an important role in cellular processes such as the regulation of gene activity, but whether and how this three-dimensional organization itself contains critical information beyond the DNA sequence was not known. In this study, we wanted to understand whether the three-dimensional folding of the genetic material could record events and exposures of a cell's past, leading to lasting changes in gene expression.
DNA is compacted by associating with specific proteins to form a structural and functional complex called chromatin. Chromatin can be regulated by epigenetic mechanisms such as histone acetylation or methylation, which modify histone proteins to control the local accessibility of every gene along the DNA. These modifications, together with the 3D organization of groups of genes contained in large chromosomal domains, contribute to determine in each cell type which genes will be active in order to produce the appropriate messenger RNAs and proteins. Therefore, epigenetic changes help organisms to produce different cell types maintained throughout life by cellular memory. To understand what molecular mechanisms contribute to cellular memory, we transiently perturbed the histone acetylation landscape of embryonic stem cells and monitored if cells could store information of this event once the perturbation was removed. Intriguingly, we found that while histone modifications and gene activity were initially reversed, chromatin folding was only partially restored. When we re-challenged the cells with a second round of perturbation, we found hundreds of genes that remained deregulated, suggesting that cells retained memory of the perturbation, and that changes in 3D chromosome structures could be the source of the memory signal. Specifically, our data indicated that the developmental repressor complexes formed by polycomb group proteins could regulate this cellular memory. Indeed, when we disrupted polycomb-dependent 3D structures without altering histone modifications, the memory response changed, and genes that previously remained inactivated returned to normal activity.
The interest of this work is that it shows how cells can record past events. We think that cellular memory is critical during development, during adult life, and in disease conditions. Additionally, studying multiple acute perturbations followed by recovery can be important in therapy, in particular in cancer, where epigenetic drugs are administered daily to patients.